now we are going to talk about photosynthesis, and we're going to go through the exact pathway that has to happen in order for that glucose molecule to get produced. You should have this document, and we're going to do a little bit of work beforehand, but mostly we're going to be on this document, and I would recommend that you be charting everything on this exact document. It'll always show up for you in red. I'll be really clear as it goes all the way through. And what I would do is in your notebook, you're going to make a list of sequential steps all the way through and exactly what happens. Before we do that, let's get ourselves oriented exactly where we need to be. Remember that we are in the chloroplast. Specifically within the chloroplast, you have all these little thylakoid stacks. Specifically within those thylakoid stacks, we care about this little molecule right here, that colorful set of stuff known as the chlorophyll and accessory pigments. That's really what we are talking about, is the chlorophyll that is found within the chloroplast on the thylakoid membrane. So that's really where this whole thing is going to get started. Okay, there's the thylakoid and then the chloroplast. Remember, the outside of both of those is still a phospholipid bilayer, which is important because we can move things across and in between those guys sometimes. We want to create there to be a hydrogen ion reservoir. We're creating that concentration gradient so we can move molecules around as needed so that we can, in the end, turn a crank, because we're all about turning a crank in this place, as well as producing that glucose molecule. And that hydrogen ion reservoir is going to be formed inside of the thylakoids. So it's really the inside, think of it like the pancakey part. That is where we make that hydrogen ion reservoir. If you are unfamiliar with that hydrogen atom and then eventually the ion, we are talking just about one proton, one neutron. Sorry, one proton, one electron, no neutrons in this guy at all. And then we're just going to be moving that one around as it needs to be. We either give it its electron or we take its electron away from it. So it just kind of depends on what we need. No electron for the ion. All right, let's jump into this, guys. So we're going to zoom in a little bit on that thylakoid membrane. Remember, this is that chlorophyll and accessory pigments spot. That's where the light's going to come in. And then we got to do a whole lot of things. So step number 1. We have we've done these. We're going to do photosystem 2. So step number 1 is that we do photosystem 2 first. It will be followed directly by photosystem 1, and really something to keep in mind is that both of these are running at the same time, but we're going to talk about photosystem 2 and then photosystem 1. They're named in that out of order fashion because of their discovery order. So photosystem 1 was discovered before photosystem 2, then we figured out that photosystem 2 technically happens before photosystem 1. Photosystem 2 has one more little component that has to happen to it in order to then trigger photosystem 1, which then triggers the last part of the process. So that's just something to keep in mind. So go grab your notebook or a piece of scrap paper or something where you can write down this list of sequential steps. But step number 1 is that light is going to hit the chlorophyll. It is going to excite some sort of an electron so that it jumps off the chlorophyll, because that is what happened when light energy truly hits an electron, is that electron gets all the energy from the light and then it goes boom and it gets excited. Remember, we're only absorbing 1 to 2% of all the solar energy that comes into a plant's thylakoid. 98 to 99% of it is just disappears. Truly, this is happening in both photosystem 2 and photosystem 1, but there's a slight difference that happens in photosystem 2, and we're going to talk about that part now. So this is the excited chlorophyll molecule. So sometimes we call that photon or that bundle of light photon. Okay, it's the same name, photon, bundle of light. We excite it, it jumps up in energy, just like when you get excited and you're all sorts of excitable. What happens is you don't stay in that excited state, just like no person stays in that excited state, no electron stays in that excited state. Eventually, it loses some of that excitability. When it loses some of that excitability, it's going to come off as heat because remember, we have to follow the conservation of mass, the first law of thermodynamics, that nothing is ever, no matter or energy is ever created or destroyed, it's just transformed. So we're going to transform light energy into excitability, into heat energy. Some light still gets re-reflected away, and eventually that electron falls back down into what's known as a ground state, or just like a relaxed state. Put number 1 right here in your diagram, because that's what's happening. It's this lightning bolt on the one side and a lightning bolt on the other side. That's what's happening. Label it light energy, pretty please. Step number 2. This excited electron is going to move down the electron transport chain. Yes, this is the exact same thing that happens in cellular respiration. We have an excited electron and it moves down the electron transport chain. 100% the exact same thing. These processes, while different, aren't all that different. Please put number 2 right here, showing an electron moving down the electron transport chain. And notice that it is going to go that way. And there's another one that is, oopsies, headed in this direction because we have photosystem 1 and photosystem 2. Step number 3. And remember, we want to really focus here on photosystem 2. A chlorophyll molecule that is now missing its electron has to have that electron replaced. It can't not have it. So that electron has to come from somewhere. The electron comes from the water. When we say that water is an important component of photosynthesis, the reason water is an important component of photosynthesis is because we're going to rip it apart. We're actually not going to use water for an H2 and an O. We're actually going to separate it into all of its components. We've got the oxygen, the hydrogen ions, and the 2 electrons that are going to get ripped away from it. So one of the reasons that we take the electron and we rip it apart is to replace the excited electron from the chlorophyll. It got real excited. Sometimes they get lost. Sometimes it doesn't just settle back down the way that we expected it to, so we replace it if it's ever lost. We replace it via the water molecule. Water is split with an enzyme. Don't worry about what its name is. Feel free to look it up if you're really curious. I'm just going to call it an enzyme. That's sufficient. The hydrogen ions that are also out there hanging around get moved into a hydrogen ion reservoir. Remember, we have to create that concentration gradient difference. And then what are you going to do with the leftover parts? Well, you're going to just let them go. And guess what? Those leftover parts are called oxygen. That's the stuff that you breathe in that gets used in cellular respiration so that you can eventually convert the glucose that we're going to make here into ATP. Super cool connected processes together. Please put number 3 over here and please write all of the parts in there. So you have H2O, we're going to split it with that enzyme, oxygen out to the air so you can breathe it in. We need a little reservoir here, and then we have just another electron that got byproduct of the process. We put it into the chlorophyll. Okay. Number 4. All that energy that is released from the electron transport chain, photosystem 2, photosystem 1, both of them, is used to actively move those hydrogen ions against the concentration gradient. So again, we're going to be using a pump in this place. And we're going from the stroma, so outside of the thylakoid in the chloroplast, but outside of the thylakoid, and we're going to pump those across the thylakoid to the inside. So like into the pancakey part. Okay. Please put number 4 right there. Right underneath number 1, right underneath the number 4. That H plus that's in red and the H plus that's here in black, those are the same one. If you want to connect those two together, that would be appropriate. Shoop. All right, number 5. We are going to take those hydrogen ions inside of the hydrogen ion reservoir. Their concentration is going to continue to build. Remember when you have a whole bunch of things that are exactly the same and they're charged? They get all, I don't know, feisty. Think if you had a whole bunch of magnets and you had all the positive charges, like you and like 6 friends, and you all tried to put a bunch of positive ends of magnets together, it doesn't stick. It doesn't work. They will repel each other and they're going to push away from each other. When they do that, they look for a place to go. The hydrogen ions in this place look to go through a channel of some kind. They want out, and they get pushed out through this little channel over here known as the hydrogen ion channel, and it so also happens to be the ATP synthase. So those hydrogen ions are rushing back out, right? They're high concentration and they are like, no, thank you. They're rushing back out, repelling each other. It's a crowded environment. Nobody likes that. We're going to follow the concentration gradient, move from high concentration to low concentration. We're rushing through that ATP synthase. This is going to be just the same as it was for cellular respiration. We're going to rush past that ATP synthase and we are going to turn that turbine. We are going to crank that turbine, that shaft around. That's got the same name because the process is 100% the same. So cellular respiration and photosynthesis at this moment both go through a thing called chemiosmosis. It's moving hydrogen ions through the ATP synthase in order to convert ADP into ATP. Okay, so please put number 6 right here and put ADP plus P, turning it into ATP. Okay, so we're going to use the same almost enzyme, coenzyme that we had earlier. Those hydrogen ions that are coming off the electron transport chain are going to find the random electrons that were bumped down the road, creating that energy necessary to pump them across the concentration gradient the way they did not want to go, pumped, active transport. And then when they go flooding back out, those electrons are just hanging out. Well, so is this NADP. NADP doesn't hang out everywhere. NADP is super smart. It knows to only hang out right around the ATP synthase. Because when it's hanging around the ATP synthase, it's going to get this influx of hydrogen ions. When it gets an influx of hydrogen ions, it's going to stick those two back together and it's going to turn into NADPH. So please put number 7 and all of its parts right here. Now we're off to stage 2. This is a great way to think of photosystem 2 and photosystem 1. This is a great way to kind of visualize those two things happening. It's really just two ladders that are taking pieces and we're literally just moving things around. Kind of like a construction site. We're just moving everything around. We're trying to get something built. It takes a lot of just moving parts around in order to get a building, a house constructed. So, must have light, must have water, must have the enzyme to split the water, and we will be constructing an NADPH, 1 ATP, and that oxygen gas byproduct. So the oxygen gas, remember, diffuses out. It's little, it's nonpolar. Get rid of that guy. We're going to take this over to number 2, NADPH and ATP. We do the light phase first because we need the light, and now we're moving to the dark cycle. Also discovered by a man named Calvin, hence why we call this the Calvin cycle. Krebs cycle for cellular respiration, Calvin cycle for photosynthesis. We're going to the stroma. We are done with the thylakoids. The thylakoids were only necessary because of the chlorophyll, so we can split, grab water, construct, and get the coenzyme that we need and the ATP. So now we're going to take both of those pieces. We're going to use the energy from the ATP. We're going to use the H from that NADPH. We're going to finally deal with the carbon dioxide that is necessary for photosynthesis. And we're going to mix them all together. Okay? We're making the glucose. Glucose is only made in the dark reaction. Okay? All right, so here's how this happens. Aren't you so excited to look at more 6 carbon molecules and how they move around. I'm going to flow you through this. Please know that I just need you to know the end products, so the reactants, and then the outputs or the products. So we're going to actually take 6 carbon dioxide molecules in this case. We're going to put them in, we got to mix them with a little thing called RUBP. It's an enzyme. Please don't worry about memorizing its name. You're more than welcome to memorize its name. I know I don't have it memorized. I just call it RUBP, and I know if I see RUBP, I'm talking about photosynthesis, and I'm talking about a plant, and I'm talking about the dark reaction. We're going to take that Rubisco. We're going to mix it with those the 6 carbons from the carbon dioxide portion of all of that, and we're going to chop it in half. If we take 6 and we split it in half, we're now going to end up with 12, because right, we had 6 CO2s. We're going to turn that into 12 what are known as PGAs. From the 6 PGAs, we're going to borrow some ATP. Thank goodness we made that earlier in the light reaction. We're going to rip that apart, get a little bit of, that's right, energy, because anytime we rip a phosphorus off of it, the end of that nucleic acid, we get a burst of energy that's released. We're going to mix it with that NADPH that we also made in the light reaction. We're going to harvest those hydrogen ions off of there. We're going to send those over here. We're going to make 12 G3Ps. You are more than welcome to pronounce that word down there. Hit the pause button and go ahead and say it. It's pronounced phosphoglyceraldehyde. Yep, super fun. Just call it 3G3P. We're going to take 3 G3Ps. We are going to use those, 2 of them, in fact, to make 1 glucose molecule. Notice you can take 3 plus 3 and you get 6. And if you're thinking, why the heck didn't you just use the 6 from beforehand? Remember how sometimes I don't tell you the whole truth and I don't show you the crazy biochemistry behind all this? You're welcome. If you're a biochemist at heart and you're like, oh, but I really want to see it, knock your socks off. Go Google the Calvin cycle. The rest of us are going to live in this world because this world is sane. Crazy Pants world is insane if you really want to go look up the molecular structure for everything. We now have 10 of the G3Ps. That's what we got left over. We need those 10. We got to recycle them because that's how we are making the RUBP. It's an enzyme, so we have to like recycle it over and over again. 10 get recycled of the G3Ps. We're going to recycle those. We're going to reuse them. We're going to borrow a few more ATPs that were made in the light reaction. That's how we reconnect and we go from G3P back to RUBP. So to do that, we have to use a little energy back in there. But that's okay, because this is how we make glucose. Remember? See? Glucose down there on the bottom? That's really all that we were after. Crazy amount of chemistry that happens inside every single thylakoid, in every single mesophyll cell, in every single plant. This is bananas. How many times we have to go through this and how many times we have to make it just to keep the plant alive. The plant does this in crazy amounts, which is how we are able to get food. We need 6 of these, again, to make that 1 glucose molecule. So, shoving it all together. This is what it actually really looks like to do photosynthesis. We take 6 carbon dioxides. We mix them with 12 NADPH2s and 18 ATPs that we created in the light reaction. We get out of that 1 glucose molecule. All that work, we got 1 monosaccharide. That's it. That's crazy. Think about how many times this has to happen. What will be left over from the ripping off of the hydrogen ions, we can create the concentration difference and we can move things around the way we need to. In that Calvin cycle are going to be 12 NADP pluses. We're going to kick those back to the light reaction so we can reload them. We've got 18 ADPs, 18 Ps. We're going to kick them back over so that we can reload those guys. And then there are 6 water molecules at the end of this that also so happen to be a byproduct. They don't actually disappear. Neither does some of the oxygen out of this guy, because we're restarting this process. Because remember, plants go through photosynthesis and then straight into cellular respiration and then back to photosynthesis and then around and around. So plants can't ever get rid of 100% of their stuff to us in cellular respiration so that they can keep themselves alive. Okay. So we're just taking the sun, we're doing a conversion. And remember, we're only converting 1 to 2% of it. 98 to 99% just goes away. We just lose it to the atmosphere. Could you imagine? What if we could capture all of that energy? What do you think that would look like? I think that would be crazy. These are your reactants. These are your products. This is the big picture that you got to keep in mind. And remember, we could really break this down and add a whole lot more to it. Okay? You need to know everything in the black. You need to know everything in the red. Where did it come from? Where is it going? What's its purpose? If you have questions, please ask them. I would print that sheet. You can print another copy for yourself. I'm happy to provide you another copy. I'm going to provide you a different version of the diagram. I would white things out. I would stick it in a sheet protector, and I would practice over and over and over again, just writing this out, writing this out, writing this out. That is how you will get it into your brain. If you just reread your notes, not going to get it into your brain. You literally need to rewrite over and over and over and over again. Okay? I promise that tactile work will help you. Go take your formatives. If you have questions, I'm happy to help you with those. Let me know what you need, and we are going to do some more modeling on this stuff. We're also running our big giant lab with algae beads so we can watch photosynthesis and cellular respiration happen all at once.